So we're starting with chapter 2, right? So we're just kind of gonna skip over chapter 1. Feel like that's one where everybody's probably already spent a good amount of time going over that. So let's get going into chapter 2. So chapter 2, and when I do SI, now, give me a second, let's see if I think my pencil might be the problem. When I do SI, I'm always gonna try to do it like kind of like how I do my notes to hopefully help direct you guys with how much detail you should do. See if I can. All right, now we're up and running. So with the cell is basically when you guys do your notes and your outlines, the cell is going to be divided into kind of 3 main parts, and then we break down each of those. So there's the plasma membrane, which remember we have a cell, right? So the plasma membrane would be the outside. So the plasma membrane is then broken down into subparts. Then we have the cytoplasm. So this would be the cytoplasm. And then the last part is the nucleus. So those are the 3 nucleus. Those would be the 3 main components of the cell, right? But then you break those down further as you start to get into more detail in the chapter, right? So like the cytoplasm, I feel like is the easiest one. So what is the cytosol, right? So the cytosol is like the fluid. So there's kind of like this jelly-like fluid. So jelly-like fluid, okay? And then the next part is inclusions. So these are kind of like extras. And I'll try to do more thorough definitions on another page, but we can probably squeeze them in. Melanin, oh, I'm gonna lose this. Okay, I will see you guys out there and I'll rewrite this when we get out there, okay? Okay, so cytoplasm, right, has the cytosol, the inclusions, and then it has the organelles. So when we kind of picture our little cell with our nucleus, right? The cytosol is kind of like the fluid, jelly-like fluid. Inclusions are extra things. So we're going to write just extra. And then we said that was melanin, carotene. So these are pigments, right? Melanin is a pigment. Carotene is a pigment. And then glycogen is the storage for glucose. So I'll draw them this way. So pigments, and then glycogen, storage form of glucose. Got it? Those are those 3 inclusions. Now for organelles, we're going to definitely need a bigger page, right? Because you guys probably remember. So there's the non-membrane bound. That would be a whole subtype with all of its offshoots, right? So we can label those. I'll just label them and I'm not going to put definitions here. Ribosomes. This is to help you guys kind of build your outline. Got it? So I know that this isn't comprehensive, okay? I'm just kind of showing you how I do an outline when I'm doing my notes. So the cytoskeleton is made up of microfilaments, intermediate filaments, and microtubules. So again, this is helping you guys build your outline. So we could actually number these, right? We could do number 1, number 2. Sometimes what I do if I'm building an outline is I'll change them up, right? I do A, B, C, and D. So C is centrosomes, D is microvilli. And then that way I can do a subgroup 1, 2, 3. Does that make sense? Or if I did 1, 2, 3, 4, I would do subgroup A, B, C. What I'm talking about is like, do you see the B cytoskeleton? The cytoskeleton has 3 components. I like to just change it up. If I'm doing numbers, I change it to letters, or vice versa. If I'm doing letters, I change it to numbers, just so that I can kind of tell that they're different. So these would all be non-membrane bound, right? And then if I'm creating again, I'm trying to create my outline here of kind of my key concepts. I have my membrane bound. So this would be my endoplasmic reticulum. Juliana, would you just put your name and student ID number in the chat? So under endoplasmic reticulum, right, then I have rough and smooth. So that would be subtypes. So I should probably keep my system going. So let's do an A, and then I'll put rough and smooth. Does that make sense? Those are subtypes of ER. And then I don't really have room, so I'm going to try to go this way. B is Golgi apparatus. And like I said, we're going to talk about each of these. I'm just doing the outline right now. Hopefully that's clear to everyone. This is not like us going through it all. Lysosomes, mitochondria. If you just came in, would you mind putting your name in the wait, okay, okay. Could you put your name in the chat if you just came in? Thank you. So mitochondria would be the fourth one, centrosome slash, then I have centrosome as membrane bound and non-membrane bound. That seems, one of those is wrong. I think that this one is wrong. I don't think the centrosome is membrane bound. And then peroxisome. We'll have to look over that. I'm not really sure. One of these doesn't belong here. Okay, so that's kind of our basic outline, but it's just of the cytoplasm. Does that make sense to everyone? Just of the cytoplasm. So what we just drew is not the whole entire cell, right? It's just the cytoplasm. So then you would go into the next part. So let's do the nucleus. So for the nucleus, you would have the nucleolus, right? And the nucleolus has a very simple function. It makes ribosomes, okay? And then the next one is the nucleoplasm. The nucleoplasm is basically kind of like the cytoplasm. It would be like the jelly-like fluid in the nucleus. Nuclear pores, those are like the pores or entrance, and we'll talk more. And then nuclear envelope. So those are your key kind of terms. Those are the key terms inside the nucleus, okay? All right, so now we go over to the plasma membrane. And for the plasma membrane, the key concepts we talk about are transport. I wonder if there's a way I could turn that sound off. Sorry if that's distracting, I don't know.
That's so loud.
I know, it's so loud, like every time it goes off, I'm like triggering my ADD. I don't know if I have ADD, but every time I'm like, maybe I do. So if you came in, just send, well, I don't know, I guess send it to everybody. Send your name and your student ID.
I know. Where's your mom works though?
And somebody has their mic on. Maybe I can, I don't know if I have the ability to mute anyone. Annalisa, maybe? I think you might have your mic on. Sorry if you have your mic on, just if you wouldn't mind muting it, just because of the background noise. So if we're talking about transport versus composition, right? So those are the two kind of types or things we're looking at in the plasma membrane. So when it comes to transport, we're going to look at passive transport across the plasma membrane. And then we're going to be looking also at active transport across the plasma membrane. So passive transport can be subcategorized. I guess I should keep my system going here. A, B, subcategorized into osmosis and then dialysis. Okay. Now, dialysis can be subcategorized into three. So this is going to be simple diffusion, facilitated diffusion, and then bulk filtration. Active transport is basically called, bulk transport is a major subtype of it. So I kind of usually write it like that because that's pretty clear. Exocytosis and endocytosis are the two major types of active transport. Endocytosis has 3 subtypes: phagocytosis, pinocytosis, and then receptor-mediated endocytosis. Okay, and then if anybody needs to leave early, remember what we said, right? Just put your name and then goodbye or something in the chat. And remember that that's just for attendance for the school's purposes. I'm not trying to control anyone. Okay, so composition of the plasma membrane. Let's do this in a different color. Well, everything else I did the same. Usually when I'm at home, I have like 15 different colors of pens. So every single subtopic would be a different color, but I don't have as many when I'm here, so I can have to keep it smaller. So composition is going to be the lipids. And then proteins. Lipids and proteins, right? So those are the two things that are composing the plasma membrane. Lipids, we have 4 subtypes: cholesterol, glycolipids, lipoprotein, and phospholipid. Proteins, we have the integral protein and the peripheral protein. Now, for the basic structure, I kind of like to add this down here under composition. The structure is just going to be phosphate and fatty acids, because that's when we look at plasma membrane, that would be the structure. Okay, so we got a lot going on the screen, right? Now you may be feeling hopefully a little bit of overwhelmed, and then maybe a little bit of clarification because you can kind of see it all at once. So the advantage to the outline format as much as possible is that it keeps your brain kind of organized and focused. So instead of getting like when you're talking here about bulk filtration, right? You're so deep into the concept that you've lost track of what you're even talking about, right? So when we talk about bulk filtration, we'll come back to the outline so we remember, okay, we're talking about passive transport across the plasma membrane. Does that make sense? So the more you refer to some type of outline, the better I think your learning is, because you can keep yourself very organized along the way, okay? So let's kind of get going. When it comes to the cell lecture, the beginning of the cell lecture, before you guys actually started talking about plasma membrane and cytoplasm and nucleus, right? You talked a little bit about the study of cells. So you talked about cytology, which was the study of cells under a microscope, right? So the study of cells under a microscope is kind of like appearance, so how they look. So for example, a red blood cell has a certain appearance. Histology is the study of tissues, right? Which you'll learn, you'll have a whole lecture for in a week or two. So the study of tissues is more like, how does this tissue look? So if we're looking at the skin, we may have different types of cells, but it's the tissue overall is the skin. So some different things that you need to know is that the red blood cell has a measurement of 7 microns, right? That's what the is. And then that the oocyte, or the female egg, has a measurement of 120 microns. So remember that when your professor is talking, something like this may seem, I don't know, may seem kind of pointless to go over, I guess you could say. It seems like maybe it's a fun thing for you guys to know and it's interesting, but these are actually going to be tested. So these are going to be on your exam. So the oocyte being the largest cell in the body, and then the 7 micron red blood cell is the smallest cell in the body. So those are all topics and things that you should know for your exam. A lot of people have been coming and going. Maybe people were here accidentally. If you're here for anatomy SI with me, Brooke, for Knudsen or Bender, please put your name into the chat with your name and your student ID number if you haven't already done. Thank you. So the different types of microscopes that are used, right, is the compound light microscope. So we're going to keep it really simple so you guys know what you need to know for the test. So it creates a 2D image. So the disadvantage is that you have to use light, and light magnifies, it magnifies in a limited way. So basically there's a limit to how much it can magnify, but it is cheap. So advantage, it's affordable. So we're going to kind of make sure that we know the basics of each one. So TEM is an abbreviation for transmission electron microscope. You probably already have that in your notes. So this is 1,000 times better resolution than the light microscope. It does also create a 2D image. It magnifies more, but the disadvantage, so this would be an advantage here. So these are both advantages, advantage. For different ones. I just didn't go for it. Yeah. So the disadvantage is that the cell or the tissue is dead. So anytime you have this two-dimensional image, this means you're looking at a slice of a tissue, right? If you look at a slide in the lab, when you guys go into lab and you see those slides, it's like a slice. You can't keep that tissue alive in order to slice it up, right? Even if it's just for those cells you see, they're dead. So any slice of a tissue is dead. So this is a disadvantage to both. So a disadvantage to the compound light microscope is that this 2D image means it's dead, right? And the same thing here, 2D image, disadvantage to TEM is that it's dead, okay? So the big advantage, it's affordable for compound light microscope, affordable. And the big advantage for TEM, it magnifies 1,000 times better than the light microscope, okay? So those are the big advantages there. All right, so the last one is the SEM, scanning electron microscope. This big advantage is that you can see the outside. So you can actually see the outside of something. So for example, if you took a needle and you looked at it, you could see the entire needle on the microscope. It produces a 3D image, okay, that's an advantage. You do not have to kill the specimen, right? Because it's looking at just the outside. So you could look at the outside of the tissue. You don't have to kill it. But the disadvantage is that you cannot see the inside. Can't see the inside. It does also magnify more than the light microscope, but we're really contrasting it to the transmission electron microscope. Okay. So that's kind of the intro, and then we just get into the cellular makeup itself, right? So like we had talked about with the outline. So this is just basically some microscope stuff, and then we get into the actual. Okay, so for the first topic, right? So we're looking here and we're on the plasma membrane. So we're going to talk about basic composition and makeup of the plasma membrane first, and then we'll go into transport next. So we're going to start with composition. So the plasma membrane is going to be our main topic. You'll probably, if you guys look at my notes, you'll see me do this. Plasma membrane is the main topic, and then my subtopic is composition, right? So now I'm going to be talking about what is the plasma membrane composed of, okay? So some basic facts, the plasma membrane is selectively permeable. Selectively permeable, that means that selective, if you're selective about the foods that you eat, right? You like certain foods and you don't like other foods. Permeable means kind of like the word passable. So what this means is the cell can choose what comes in and goes out. So, you know, if the cell doesn't want potassium, it doesn't need to let potassium in. If it does want potassium, it can let potassium in. Does that make sense? So the cell chooses what comes in and what goes out. So composition, the plasma membrane is also called mostly the phospholipid bilayer. I hesitate to say that since technically the phospholipid bilayer isn't all of the plasma membrane, but they're 99% the same. So we can basically say they're the same thing. So the phospholipid bilayer, this is the major component of the plasma membrane. Like I said, it's composing like 99%. It is made, this is a phospholipids, right? So phospholipid is a phosphate head and a lipid tail. So it looks like this. Phosphate head, this is phosphate. And then lipid, let's draw them in yellow. Phosphate head and lipid tails. So what happens is you have your cell, right? And then you have all of these little phosphate heads with lipid tails. I'll try to explain this. I'm going to try to explain it in the way that you need to know, especially for moving on to physiology, but try not to get it too complicated, okay? So extracellular environment is mostly water, okay? So this water means hydro, right? So hydro, like a hydro flask, right? Hydro flask is like a water jug. So hydro means water. Intracellular, the intracellular environment is the inside of the cell. The extracellular is outside the cell. Both of them are mostly water, okay? So what do you know about fats? So lipid tail is made of fat. It's a fatty acid tail. Don't fats repel or hate water? Like if you pour oil, like olive oil, into a glass of water, doesn't it hate it? Like it forms balls and all rises to the top. It doesn't mix with the water, right? Oil. So the lipid or fatty acid tails don't really like the extracellular or the intracellular environment. So that is why you end up with a phospholipid bilayer, because the phosphate heads love the water. So they're like attracted to the water. And then all the little lipid tails turn inward away from the water. So you kind of end up with this bilayer. So it looks like this with extracellular and the phosphate heads being attracted to the extracellular, and then intracellular with the phosphate heads attracted. And then those fatty acid tails are trying to stay away. And we call it bilayer because there's two sets. Does that make sense? There's one row on top, and then there's a second row on the bottom. So both of the head part is loving or moving towards the water inside the cell and outside the cell. But the fatty acid tail is staying away from the water. And there's two layers, which is why we call it a bilayer. Okay, so let me write out. Everybody at this point is like, okay, well, what do I need to know for the test? Like, that's kind of confusing. So that's the overall topic. So what you would want to know is you would want to know that phosphate heads are hydrophilic. The word philic here means water loving. Okay, so hydrophilic, water loving. So the phosphate heads are water loving. Another word of saying hydrophilic is polar. So they're polar, they're attracted to the water. So all of this, would be what you would want to know for the test. So what about fatty acid tails? Fatty acid tails are also called lipids, right? They're a type of lipid. We'll get into that. There are 2 per phosphate head. And these are non-polar and they're hydrophobic. Kind of like if you have a phobia of something, you hate it. So they hate water, the fatty acid tails. So this is the simplest way of saying what you need to know for the exam. But hopefully you understand the concept so that you guys can apply it a little more. But that's the simplest way of saying it, okay? Sometimes memorizing is easier if you get the concept. Okay, any questions there? All right, so for the next part of the plasma membrane, we're still talking about composition, and now we're going to be talking about the lipids. So let's go back to the outline real quick and I'll show you to remind you. So the first one is lipids. So if we look at our little outline, right, we're right here, lipids. So when we look at lipids, which lipids are found in the plasma membrane? So first, we have our phospholipids. I just told you what those are, right? Phosphate head, and then the 2 fatty acid tail. Phospholipids, those are this would also be something you want to know for the test. They are the most common structure in the plasma membrane, right? And because they are a bilayer, the plasma membrane is a double membrane, right? Okay, so the next thing, glycolipid. So the glycolipid, the function of that is for cell to cell recognition, okay? And again, to remind everybody, whenever we go over stuff in SI, I'm not gonna write down, I'm not gonna waste your time or my time writing things down that are not on the test. So if I go over it and I write it down, it's pretty much safe to say, well, not pretty much, it's on your test. It's more like there's things I can't get to that are on your test, then I'm gonna waste your time with stuff that's not on the test. Does that make sense? So if you're spending your time here, we're only going over stuff that you're going to be tested on. We're not wasting time with other things. So, for example, what is on the test from here? You guys need to know what are the 4 types of lipids that are found in the plasma membrane and what are their basic functions. So, phospholipids, most common structure in the plasma membrane. You guys need to know what we just talked about, right? What are the phosphate heads? What are the fatty acid tails? Now, when we talk about the next type of lipid, what is the glycolipid? It functions for cell-to-cell recognition, and 1 fatty acid is replaced by a carbohydrate. So this is kind of getting more into molecular stuff. So really, I'm just going to erase this because honestly, that's not how he'll test you. He wants you to know what are the 4 lipids and what are the 4 functions. So let's keep it with that. So glycolipids, cell-to-cell recognition. Lipoprotein holds cells to one another. And then cholesterol, this one holds the phospholipid bilayer together. So if you kind of picture what we had talked about, right, with our little phospholipids and then their lipid, right, their lipid tails, did it seem like anything was really holding them together? No, right? It was just that they loved the water on the outside and then they loved the water on the inside, right? So to hold them together and actually come through like a glue is the cholesterol, okay? So the cholesterol is holding that bilayer lipid together, keeping it all secure, okay? So lipoprotein holds cells to one another, forming tissue, and then glycolipid also for cell-to-cell recognition, helping form tissue. So those are 4 lipids of the plasma membrane. Now we do plasma membrane. We're still on composition. And now for composition, the next thing we're doing is proteins, right? So we kind of had two big subtypes. Oops. Protein and then our lipids. So for protein, we have integral protein and peripheral protein. So those are the two types of protein. So proteins, just a few little sub details here. Proteins are made up of amino acid, okay? Proteins are what make the plasma membrane selectively permeable. So remember how we talked about the plasma membrane is selectively permeable. Certain things can come into the cell and certain things can't. So that's because of proteins. So we're going to talk about how things get transported. So the integral protein is also called the transmembrane protein. And the peripheral protein does not have another name. Okay, so we're going to make sure we have two names for integral protein. So the integral protein allows large or charged compounds to enter the cell. Large or charged compounds enter the cell through the integral protein. The peripheral protein is attached to the integral protein. It can either be attached on the outside or the inside. So we'll write on outside or inside. And its function is to make the integral protein channel more specific, okay? So basically, let's give an example. So let's say glucose wants to come into our cell. So I'm going to try to draw a little cell here so that we can kind of picture it. I like to draw things out. So we have our plasma membrane, right? I won't draw it all the way, but we kind of talked about our little phospholipid bilayer. So phospholipid bilayer is kind of all through here, yeah? Now our integral protein looks something like this. It's kind of like this channel. It has like a gateway through it. And through that gateway, glucose, this is our glucose can fit. So if you picture it, anything that can fit just comes through. Does that make sense? If it's an integral protein, anything that can fit can pass through and come into the cell. So in this scenario, glucose is coming into the cell, okay? Now, if the peripheral protein is present, like let's draw a little peripheral protein, we'll kind of imagine it being present. It's going to create like a lock and key mechanism. So let's kind of draw a little shape. So if this peripheral protein is here, the glucose cannot come inside the cell, right? Unless it can fit into the lock. So let's say we had, give a good example. Let's say we had potassium, and potassium walks right up to this cell and says, okay, I'm ready to come inside. If potassium wants to come in and it can't fit, it won't be able to enter the cell. Okay, so if potassium looks like this, it can't fit onto that channel. Can't get in. So the peripheral protein makes the channel more specific, like a key for a door. So I'm putting that in parentheses because that's not like how it's going to be on your test, but it's to help you understand it. So it's like a locking mechanism so that things specifically can get in. Again, an integral protein will allow any large or charged compound to get into the cell, but if we add a peripheral protein, it makes the channel more specific. So the example in your body of a peripheral protein, this is the most common kind of type, is an enzyme. So an enzyme is your key example. So enzymes make the cell more specific. so that only certain things can get into and go out of the cell. So just to be clear on that, we could also have a peripheral protein on the inside so that potassium is not allowed to leave unless it fits into that little key. Is that making sense to everyone? Right, so things have to fit into that special little lock in order to leave in this case.
Brooke, can you show the last slide really quick so I can get a picture of it? And thank you so much.
Yeah, no problem. Okay, so let's go back to our little outline, do some check marks. So we talked about lipids, right? We talked about cholesterol, glycolipids, lipoprotein, phospholipid. We talked about protein. We talked about the integral protein and the peripheral protein. So I'm going to kind of add a little plus box here and put the word glycocalyx, okay? So glycocalyx is kind of in this area because it's attached to the integral protein. So it's another structure that you guys need to know about, but it's not technically a protein. Does this make sense? So it's not a protein, it's more like it's attached. So when we talk about it, I just want to make sure you're understanding it's not a protein. So when we talk about the integral protein, right, attached to our integral protein is our glycocalyx. So we can kind of have 3 examples of the glycocalyx. So we'll draw 3 colors so that we can kind of make them stand out. So if you imagine 3 little arms coming off of our little IP, our integral protein. So the red would be our, well, so let's say that this is our, well, my color is not cooperating. Glycolipid. This is our first glycocalyx example. Okay, so a glycocalyx is basically a tree limb attached to the integral protein, and it has a variety of functions. So I'm going to write down the functions of each one. So if it was a glycolipid, this would function for cell-to-cell recognition, adhesion, and communication. You may be thinking, wait, that feels familiar. I feel like I just wrote that on my paper like 2 seconds ago. This is the same glycolipid that we talked about right here. So the glycolipid that is a lipid found in the plasma membrane is one of the members, we'll just write the word members of the glycocalyx. Okay, you'll see when I get to it, the next one is going to be lipoprotein. Okay, so we'll go back over here. So the next one is lipoprotein. So if it was a lipoprotein, again, another member of the glycocalyx, same function as we already wrote down, but I'm just going to write it one more time. It holds cells together to form tissue. Again, we already talked about this. These both are lipids that are part of the plasma membrane. But the third one, the black one we're pretending, is called the glycoprotein. And we haven't talked about glycoprotein yet. So glycoprotein, this is for cohesion, communication, reorganization. Okay, so how do you differentiate this for your study? So you need to know that there are 3 types of glycocalyx, right? You need to know that there are 4 examples of lipids, right? And then you would know their functions, which I feel like is self-explanatory. So if the question is asking you about the glycocalyx, you would know glycolipid, lipoprotein, glycoprotein. But if the question is asking you about lipids, you would know phospholipids, glycolipid, lipoprotein, cholesterol. If the question is asking you what is a glycolipid, you would know a glycolipid is part of the glycocalyx and it's a lipid. A lipoprotein is part of the glycocalyx and it's a lipid. Does that make sense? Versus a glycoprotein is just part of the glycoproteins. Okay, so I'm not trying to confuse you. I just want to make sure you realize they're kind of classified 2 areas. So that's why sometimes students get confused by what they are. Someone asked something here in the chat. If you asked me something, sorry, I'm not sure exactly what you said. Oops. Oh no. Oops. No. It's very sad. I just lost my whole outline. Okay. I'll have to redraw it. So I lost my outline, but we'll keep going. So basically we had gone through the part of the chapter that we talked about the plasma membrane. And then we talked about our lipids and our proteins. Yes? So we had done 1 and 2. So that was the composition. So now we'll do transport. Typically transport takes a good while. I usually will just tell you guys that because sometimes I know that when it starts to get really detailed, maybe you get discouraged, but the transport to me is the most challenging part of the chapter. So once we get through transport, it does start to get better from there. So I want to just encourage you that that is the most challenging part. So for passive transport or diffusion, that's going to be our first topic. And we're going to look into osmosis and then dialysis. And dialysis, there are 3 subtypes of dialysis: simple diffusion, facilitated diffusion, and then bulk filtration. Those are the 3 subtypes. Okay, and we'll just do 1 and 2. And then for active transport, right, we had exo and endo. And we'll go into the 3 subtypes. Okay, so as we go through that, just a reminder that we will get through transport and then it kind of gets a little better from there. Things get a little more simple. So for transport across the plasma membrane. So when we kind of picture, just try to keep that picture in your head of that cell. And remember that when we talk about a cell, it's made up of those phospholipids. And then what was kind of in between is the integral protein, right? Also called the transmembrane protein, allowing things in and out. So kind of keep that structure in mind. Because
Brooke, I have a quick question.
Yeah.
Do those tails touch ever or no?
They actually, that's a great question. They actually float. So they're just floating. And that's why the cholesterol is so important, because without cholesterol, it's almost like they have a tendency to just float away to maybe another cell. So it's like they just be like, oh, I'll just go wander over here. I'll wander over here. But because of cholesterol being in place, it does kind of keep them together. But no, they're not attached to anything. They're just kind of waving in the breeze or waving in the cytosol.
Okay, thank you.
Yeah. So transport. Our first type is osmosis. Okay. Osmosis. So for osmosis, this is the movement of water. Okay, so the movement of water. So osmosis, or the movement of water, this moves along with oncotic pressure. So it moves from a high, moves from an area of high oncotic pressure to an area of low oncotic pressure. Okay, so osmosis, movement of water, it's moving along with the oncotic pressure from an area of high oncotic pressure to an area of low oncotic pressure. And remember, this is our first example of, I didn't write this as my heading. This is our first example of passive transport across the plasma membrane. So when we talk about passive transport across the plasma membrane, we are talking about always, these are always examples of transport that do not always require no ATP. So they never require ATP. That's the big thing with them. And then the second rule for passive transport is they always move down the concentration gradient. So that's why for osmosis, we make sure that we tell you that oncotic pressure is the specific word we use for water, but osmosis is moving from high to low oncotic pressure, okay? Trying to keep this as simple as possible. Just basically, oncotic pressure is a very complicated concept, but water moves from high to low oncotic pressure, okay? It has to do with the dilution of solutes. So it's saying like, if there was a lot of salt in a solution, water would want to basically move to dilute that salt. It's just kind of complicated for this class, so we try to make it simple. So just know high to low oncotic pressure. With dialysis,
Sorry, really
yeah,
quick. So when I wrote the notes for osmosis, in the PowerPoint that he has, he wrote it as osmotic pressure. I don't think I noticed oncotic. I'm just
yes,
wondering.
osmotic and oncotic are the same. Let me rewrite it.
Okay.
Sorry, I was doing I think that my chemistry brain picked in. Osmotic or oncotic would be the same. So we'll do osmotic. Sorry. Yes.
Gotcha. Okay, thank you.
Yeah. So dialysis, good catch there. I was switching over to too complicated. So dialysis is the movement of solutes. So we had movement of water, movement of solutes. So if we picture a glass of water, and then you know that the hydration multipliers or Gatorade or something or salt, if you picture all those little like particles in the water, those would be our solutes versus the water is just surrounding it. Does that make sense? So solutes are like salt. Wow, I don't know why I had an H there. Salt, potassium. So these are like natural solutes. Magnesium. There are multiple, those are just the ones I'm thinking of off the top of my head, but we'll write etc. Okay, there's a lot of examples of solutes. So simple diffusion or dialysis, simple dialysis, this one also called dialysis, right? Simple diffusion, simple dialysis. This is the first example. This in simple diffusion, no integral protein is required for the transport of these solutes across the plasma membrane. So remember, we're talking about transport, we're talking about things moving across the plasma membrane. The movement of water makes sense, right? I just have water out here, water gets inside, okay? But the movement of solutes is a little bit more complicated because we're talking about solutes now and not just water. In simple diffusion or simple dialysis, something just crosses the plasma membrane based off concentration gradient. So what that means is if we have a high concentration outside the cell, so let's say this is potassium molecules and we have like so much potassium out here and there's only a few, we're going to move from the area of high to low concentration of potassium. High to low potassium. K plus is the abbreviation of potassium. So high amount to low amount. High to low is moving down or with the concentration gradient. So that is what governs simple diffusion. Simple diffusion just passes through the phospholipids. So remember how I said the phospholipids are kind of just floating, they're kind of just hanging out. There are little tiny gaps or spaces in between the phospholipids, right? And that those little tiny spaces allow passage of things in between them. So if you picture, here's my phospholipid here, and my lipid tails here. Anything that can fit or wiggle its way through that gap can pass through. And that would be simple diffusion or dialysis, because it's just going to move from high to low. So let's say we had a lot of sodium. This cell has so much sodium, it's like overloaded with sodium. And we have a little tiny bit of sodium out here, just a little bit. Because of simple diffusion or simple dialysis, it will just pass through or move through the phospholipid bilayer from inside to outside. So does that make sense? Dialysis is the movement of solutes from inside to outside or outside to inside. It doesn't matter. It's just always requiring no ATP energy and always moving down the concentration gradient. So do you guys see how it doesn't require energy? It wants to go that way, right? If I'm overloaded with sodium, I want to get rid of it, right? If I don't have any potassium, I want potassium, okay? So that was the first example of dialysis. So we talked about passive transport. We covered the first example, which is osmosis for water. And then the second is dialysis. And dialysis is for the movement of solutes, right? And what are the two big rules with passive transport? Requires no ATP, which is energy, no cellular energy, and it moves with or down the concentration gradient, right? So our first example is dialysis. Then dialysis has simple diffusion. And we said this requires no integral protein. It's just infusing across the plasma membrane with just goes through, I'm going to write PL, just goes through the PL bilayer. But for facilitated diffusion, which is our next example of dialysis, this one requires the integral protein. So right away, you're probably thinking, okay, well, if it requires that integral protein, it probably means it's too big to get through just the plasma membrane, right? So we also call this integral protein a carrier molecule or a carrier protein. So the carrier molecule or the carrier protein, this is what allows facilitated diffusion to happen. So maybe we have glucose, so examples, large, polar, hydrophilic. Oh, sorry, I don't know what happened to my spelling there. Hydrophilic. So large polar hydrophilic molecules cannot fit through the phospholipids. So these large polar hydrophilic molecules, they must use an integral protein. So the only way they can get into the cell or leave the cell to go out, they need to use an integral protein. So let's say this is my glucose molecule. It's going to go through that integral protein. It's still going to go from an area of high concentration to an area of low concentration. So for example, if this is my cell, if I have a lot of glucose here, glucose, and I only have a little bit of glucose here, it's still following the same exact concept of before, going from high amount to low amount, but the only difference is it's going to use that integral protein. Does that make sense? Same concept as simple diffusion, it's just that it uses the integral protein for large polar or hydrophilic molecules. Okay, some examples that you would need to know for simple diffusion of what can pass through is oxygen, carbon dioxide, okay, and non-charged molecules. So if it's not charged, it can pass through. Good. Our last example of passive transport is very simple. It's just called bulk filtration. And there's not a whole lot you need to be aware of for the test. Just know this allows large amounts of substance to pass through. Okay, so we have what? We have 3 examples of dialysis. Are these 3 examples of passive transport? No, right? We have 1 example of passive transport, which is osmosis or the movement of water. Second example of passive transport is dialysis or the movement of solutes. It has 3 subtypes: simple, facilitated, and bulk filtration and diffusion, right? So now we move on to active transport. So let's do a different color here. Let's do black. So for active transport, you're already seeing the word active, right? So the key here is that this requires cellular energy, or ATP. This is because the cell is doing something it doesn't want to do. It is moving against or opposite or up the concentration gradient. So whereas passive moved from high to low, this is going to move from low to high concentration. So you're going to force the cell to do something it doesn't want to do, right? If there's a lot of glucose here, right? Lots of glucose already, and there's only a little bit of glucose here, it doesn't matter. We're still going to force that tiny little bit to join the big amount. We're going to go from low to high, forcing, kind of like peer pressure, right? We're forcing the cell to do something it does not want to do. So this is it needs 3 things, right? It needs to go against the concentration gradient. It needs energy. And then it also requires a transport protein. So the transport protein is much like the integral protein, just specifically called transport if we're talking about active. So we need an integral protein. Okay, so there are 2 types. So we have active transport does have a lot of different subtypes, but the main subtype we talk about in anatomy is called bulk transport. Bulk transport refers to bulking up substances into vesicles. So we're saying that we're transporting substances. Let's just replace that with the word transporting. Transporting substances in vesicles. So these are little like kind of bags or sacks. So the first example that you guys learn about is exocytosis. Okay, exocytosis. So during the process of exocytosis, a substance exits the cell, right? That kind of makes sense. Exocytosis, a substance exits the cell through a secretory vesicle. So something is being secreted or sent out of the cell through a little vesicle or a package. Okay. B is endocytosis. Endocytosis is the opposite of exocytosis. So a substance comes in to the cell through a vesicle from basically pinching the plasma membrane. It's grabbing the plasma membrane out. So it's like the cell forms arms and pulls something into it in endocytosis. Okay, so let's do a little more about endo. And I can go back if you guys need me to go back. I know sometimes I write a little bit fast. So the first type of endocytosis is phagocytosis. Sorry, somebody asked for the microscope picture. I accidentally deleted that one. I can go through those again, basically the compound light microscope. I should have a picture. I can send you guys a picture as well of that page if you need it. So endocytosis, again, we talked already about exocytosis, right? Remember, you guys, if you need to leave early, sorry to repeat it. If you need to leave early, please just put your name in the chat box, right? Saying that you needed to leave early, just so that I know. So phagocytosis is when the cell brings something in, cell brings a substance inside, okay? So it forms arms, and the arms are called pseudopodia, and those arms kind of bring the substance in, okay? So if it's phagocytosis, white blood cells do this. So white blood cells actually eat bacteria or parasites and bring it in. So if you picture your little cell here, pseudopodia are kind of like this, like these little arms. These are pseudopodia. And here's our little bacteria. And with phagocytosis, it's going to eat that bacteria, bringing it in, and then it's going to kind of envelop it and put it into the cell. So for a white blood cell, that would be so the white blood cell can kill it, right? Next one is pinocytosis. So this is the same concept, but the cell brings a liquid inside. And this is cell drinking. Phago means to eat. This is cell eating for phago. Pinocytosis is cell drinking. Easy way to remember it is like a pina colada, right? Is like a drink. So just think of it like that. You're drinking something. Receptor mediated endocytosis is the third type. And this is like the other ones, but this requires a protein channel with a receptor site so that only specific cells, only specific substances come inside the cell. Okay, so for receptor mediated, it's still endocytosis, but it's requiring specific things to come into the cell. All right, so that would be everything for transport across the plasma membrane. So not too bad, right? So we covered exocytosis, endocytosis, we did phago, receptor mediated endocytosis. That's everything for transport. So now we have the cytoplasm, right? So we're going to go over the cytoplasm. which consisted of the cytosol, the inclusions, and then the organelles. Hopefully, if we can, we'll get through cytoplasm and then either start nucleus or finish nucleus, but that will leave us with a good area to go into for mitosis on Thursday, right? Because for mitosis, we need to talk about the nucleolus, the DNA, and then mitosis, okay? So definitely remember, I'm not gonna, if you guys need to leave, that's fine. I just always remind you guys, I won't be able to go back over anything. Okay, so for the cytoplasm, right? First area of cytoplasm, cytosol. If you're looking at the cytosol, what is it? So the cytosol is basically the jelly-like fluid in the cell. We've spent this whole entire time talking just about the plasma membrane, right? Just about that outside portion of the cell. That whole time we talked about that. That's because it's kind of the most complicated. Now we're talking about the little cytoplasm, the stuff that's kind of inside the cell. And then the last area is the nucleus. Does that make sense? Kind of the three portions. So for the cytosol, it's all that jelly-like fluid that's holding up the cell. It has a variety of things in it. It has ions. Ions are charged, basically charged substances. So for example, sodium, potassium, chloride, right? These all have little charges on them if you've taken chemistry. Nutrients, examples would be amino acids, monosaccharides. Some of these words are unfamiliar, but you'll learn them as we go along. Proteins, example of this would be the cytoskeleton. Carbs, okay, lipids, and other small molecules. So you would need to know all of those for the test, right? That ions, nutrients, proteins, carbs, lipids, and other small molecules. You need to know that all of that is found in the cytosol of the cytoplasm of the cell. The next thing that's found in the cytoplasm of the cell, and let's use a different color so it kind of sticks out, is the inclusions, right? So the inclusion. These are temporary storage for complex molecules found in the cytosol. Okay, so we have melanin. Melanin is black pigment. Okay, so when we tan, right? When a white person tans, that's like black pigment. Every area of the world has a different variety of how much because it's a natural sun protector, right? So the lighter you are, the less sun exposure that people group had, and then the darker would be more sun exposure. So it's basically a UV protectant, which I think is really cool. It's like natural sunscreen. So that's what melanin is, okay? So it's for a UV protectant. Same thing for yellow orange pigment. This would also be kind of like a UV protectant, okay? So that's just kind of the biology of what that looks like. And then glycogen, okay? Glycogen are long chains of sugars, okay? So these are from stored in the liver and the skeletal muscle cells. So cells in the liver, cells in the skeletal muscle store glycogen, and glycogen is the storage form of glucose. So if you are starving, right, what your body does is it breaks down glycogen so that you can have access to glucose. Glucose is the only thing that your neurons run on. So your brain only runs on glucose. So glycogen would be the storage form of glucose. So for the test, right, you could be asked, what is glycogen? You could also be asked, where is glycogen found in the cell? And you need to know that it's an inclusion in the cytoplasm. So that's cytosol and inclusion. So if we go back to our little diagram, we did cytosol and inclusions. Remember, organelles is going to get complicated, right? Because we need to do non-membrane bound and membrane bound. Okay, so that's what we're going to talk about next. So everybody with me, cytoplasm, cytosol, cytoplasm inclusions. Any questions at all? As always, if you guys have questions as we go along, it is helpful. If you ask them, I think if you're confused about something, always let me know, because I think other people are too. You're welcome to put those in the chat or raise your hand. I think that it kind of notifies me pretty well if you raise your hand. So if we do organelles, we're going to start with membrane bound. So if something is membrane bound, each of these organelles that are membrane bound, right? They have a phospholipid bilayer. So remember how we talked about that little phospholipid bilayer, right? Each of these little organelles within the cell has one. So picture your cell, right? And we said it has a little phospholipid bilayer going all the way around it, right? Now, inside of the cytoplasm of this cell, you have other little things that have their own. So let's do an example. So maybe we have our nucleus, and then attached to the nucleus is kind of this rough endoplasmic reticulum. Now it has its own little phospholipid bilayer on it. Does that make sense? So each little guy, maybe if I do it the right color, each of these little organelles would have its own phospholipid bilayer. So the first membrane-bound organelle is the endoplasmic reticulum. So for endoplasmic reticulum, you want to memorize this definition. A series of tubes and channels for the transport of proteins and other substances. Okay, so our definition of the endoplasmic reticulum is a series of tubes and channels for the transport of proteins and other substances. Now there are two examples, right? Two examples. There's rough.
Brooke,
Yeah.
I have a quick question. So it says a series of tubes or tubules.
Oh, sorry, tubes.
Okay, thanks.
No, you're good. My handwriting on the iPad, it's always the worst form of my handwriting. So if anybody can't read anything, please let me know. I know that I write fast and sometimes things don't come out well. Thank you. So if it's rough endoplasmic reticulum, this means it has ribosomes attached. Okay. If it's smooth, it does not have ribosomes attached. Right? And you're thinking, oh, great, that's the definition. And sadly, that is not the definition. That's just part of the definition. So if it's rough, it has ribosomes attached. The function of rough endoplasmic reticulum is to transport proteins. Why? Ribosomes make proteins. So if we picture, let's draw it a little bit bigger so we can see it. If this is my endoplasmic reticulum, right? And these are my little, on the surface, these are my little ribosomes. The ribosomes are going to be pumping out or creating little proteins, and the proteins are going to go right in the middle of this tube, okay? And that endoplasmic reticulum is this tube, series of tubes and channels that is going to transport that protein wherever it needs to go. Does that make sense? So again, this is the nucleus, and those ribosomes are sitting out here on the edge of my rough endoplasmic reticulum. They're making protein, and the rough endoplasmic reticulum is transporting proteins made by ribosomes. Okay, those proteins will then be shipped out of the cell. Okay, so those proteins will then be shipped out of the cell. Does that make sense? So they're on their way out. Smooth endoplasmic reticulum has a different function, right? Because it doesn't have those ribosomes attached. So it is going to function for synthesis, transport, and storage of lipids. That would be function number 1. Function number 2, trying to draw a little line here. So function number 2 of smooth endoplasmic reticulum is to metabolize carbs. Function number 3 of smooth endoplasmic reticulum is to detoxify drugs, alcohol, and poisons. All right, so smooth endoplasmic reticulum does not have ribosomes. It's functioning for a whole nother thing. It is working on lipids, carbs, and drugs, alcohol, and poisons. And it doesn't have any ribosomes attached, right? Okay, so this was all that we talked about this whole entire time. We didn't talk about membrane-bound organelles, right? We just talked about one membrane-bound organelle, the endoplasmic reticulum. And we explained that there's 2 subtypes, and we talked about those. So if you go back to your little outline, let's add ER, rough and smooth. And we'll add that as our number 1, our first one we talked about for membrane-bound organelles. So what's our second one? Our second example of our membrane-bound organelle, right? So these are organelles. These are all in the cytoplasm. These are the membrane-bound ones specifically. And our next example is the Golgi apparatus. All right. So remember, we have our little nucleus. We have our cell, our nucleus, our rough endoplasmic reticulum transporting those proteins, right? And those we said we're going to leave the cell. Well, the Golgi is the key to getting them to leave. So let me put that Golgi in black. So Golgi apparatus kind of looks like this. It's connected to the rough ER. And this is the Golgi. So the Golgi receives proteins and lipids from the rough/smooth ER. So some parts of the endoplasmic reticulum may have ribosomes attached. And if it has ribosomes attached, what did we say it was called? Rough, right? Rough is when it has the ribosomes. Some portions may not have any ribosomes, and we said that that was smooth, right? So what it looks like is it kind of looks like this. It keeps winding, and parts of it have ribosomes, and they're called rough. And then parts of it don't, and they're called smooth. So if it's the part that has ribosomes, what is it transporting? Proteins. If it's the part that doesn't, what is it transporting? Lipids. So once it gets over here to the Golgi, right, and the Golgi receives, the Golgi is going to receive proteins and lipids. So that's why we say receives proteins and lipids. And it modifies, packages, and secretes them out of the cell into secretory vesicles. So that's how they leave the cell. They're going to leave the cell as a secretory vesicle, or they stay inside the cell as a lysosome. So if those proteins and lipids leave the cell, so maybe it's through these little secretory vesicles, they're going to be called a secretory vesicle. But if that same little vesicle ends up staying inside, we call it a lysosome. Okay. There is a receiving region of the Golgi, and that is called the cis face. So it's the part that receives from the ER. There's a shipping region. So Golgi is kind of like your UPS store. Or your Amazon store. There's a shipping region, and that's called the transface. Okay, so the shipping region, the receiving region is cis, so it would be this side, and trans ships it out. Cis, trans. All right. So that's the big function of the Golgi. The Golgi is pretty important, has quite a few functions, right? Secretory vesicles is what the name of it, if it packages it out. Lysosome if it stays in. All right, so what's our next membrane-bound organelle? So that was membrane-bound organelle number 2. So going back over here.
I have a question.
Yeah.
What did it say before the word modifies? I can't really read the handwriting.
Oh, sorry. This is supposed to be and it modifies, packages, and secretes.
Thank you.
So the next one is actually lysosome, right? So our next membrane-bound organelle is the lysosome. So again, where does the lysosome actually come from? It comes from the Golgi. So what is its function? So membrane-bound, these are organelles in the cytoplasm, right? Organelles. This one is the third type we've talked about. It's the lysosome. And where does it come from? Comes from the Golgi apparatus, and it's from things that were packaged vesicle that stayed inside the cell, right? It is a waste management organelle. It functions for intracellular digestion, okay? It contains enzymes that break down products in the cell. So basically, like, let's say you went through a metabolic process and created waste. You know how they say, like, you have free, there's actually free, like, hydrogen ions, or people say you detox, right? Like when you detox. So you detox, these lysosomes are detoxing all the time. If they detox to kill the cell, right? That's different than the automatic normal process. So autophagy is automatic eating by the cell of damaged organelles or waste products. So basically your lysosome is constantly going around. If this is my cell, here's my nucleus. That little lysosome is just on its way, kind of like a little Pac-Man, right? And it's eating up little waste products in the cell. Does that make sense? That would be called autophagy. Autophagy is the process. Autophagy is done by the lysosome. But we also have something, I'll write it kind of in red. This is called autolysis. And autolysis is different. Autolysis is when these same enzymes dissolve the entire cell. So if it was a cancer cell, right? So let's say that the cancer invaded that cell, your body would try to protect you and kill off that cell, right? So this occurs after autophagy. So maybe your cell tries its best to kill off the bacteria or the cancer that enters, but it just can't get a hold on it. And in order to protect you, it kills itself, right? Kind of like, I think they call it kamikaze, right? So it goes down with the fight. It kills itself. It self-destructs. That's called autolysis. Autolysis, it self-destructs. So that's the third membrane-bound organelle. Good. So we had first membrane-bound organelle, endoplasmic reticulum. Second membrane-bound organelle is the Golgi apparatus. Third membrane-bound organelle is the lysosome. So the fourth membrane-bound organelle is
Brooke, would you mind going back to the Golgi apparatus, please?
yeah, sure. There we go.
Thank you.
No problem. Okay, so we have our membrane-bound mitochondria. This is our one that most of us have heard of, right? So mitochondria, the function of mitochondria is to create ATP, which is cellular energy. And it does this through a process called cellular respiration. Okay. Cellular respiration, again, is creating our cellular energy. This occurs on the crista on the matrix of the mitochondria. So our mitochondria kind of looks like this, and it has inside matrix. Matrix increases surface area. That would be something you'd want to know for your test, right? Matrix increases surface area. And then lining the matrix are these little crista, which are balls, right? I think of them like little crystal balls. So balls where ATP is made. So those crista are actually making the ATP through cellular respiration. The other really important thing about the mitochondria, it is double membrane. Okay, so the three double membrane structures are the nucleus, the mitochondria, and the plasma membrane, right? So three double membrane structures are the nucleus, mitochondria, and plasma membrane. All right. Any questions? All right, so mitochondria is our fourth membrane-bound structure in the cell. So this is definitely a test question here, that there are three double membrane structures: the plasma membrane, the nucleus, and then the mitochondria. All right, so our membrane-bound, I believe we have one more here, and that is our peroxisome. Hang tight, we've only got 10 more minutes and then we'll resume on Thursday. So our peroxisome, these function threefold. They detoxify, they break down fatty acids, amino acids, and poisons. Okay, and then they convert hydrogen peroxide to water. So 3 functions of a peroxisome. They're detoxifying, they're breaking down fatty acids, amino acids, and poisons, and then they're converting hydrogen peroxide to water. So those would be the 3 ones that you would be tested on. Okay? So that's everything for the membrane bound, right? So that is everything for this membrane bound section. So then we do non-membrane bound, and then we finish cytoplasm, right? So this is why it kind of helps to look back at the outline. We've already done composition and transport of the plasma membrane, cytosol inclusions, and we're almost done with organelles. And then this way on Thursday, hopefully I'm hoping to start here with the nucleus and add on mitosis. Good?
Brooke, can you go back to that slide really quick, please?
Absolutely. Yeah.
Okay, perfect. Thanks so much.
Yeah. And if anybody's missing any, I'm happy to go back to those as well. I don't mean to go move on too quick. Always let me know if you need me to go back. So non-membrane bound organelles, these do not have a membrane around them. Our first is our ribosomes. Remember, we already kind of talked about them, right? They are the site of protein synthesis. So where protein gets created, remember, they are attached to the rough ER, right? So we call them fixed if they are attached to the rough ER. We call them free if they float in the cytosol. Okay, so if they're attached to the rough ER, they produce proteins that leave the cell, right? Because they went to the Golgi, they went to the Golgi, and then the Golgi shipped them out. If they float in the cytosol, they produce proteins that stay, or they could be shipped out, okay, but often stay.
Bro, please, can you quickly go back to where you have the Golgi, the slide?
Yeah.
All right, thank you.
Yeah. And the ribosomes are the most abundant organelle. So there are the most number of ribosomes out of all of the organelles. The next non-membrane bound organelle is the cytoskeleton. And the cytoskeleton has 3 components. It has the microfilaments, the intermediate filaments, and then the microtubules. So those would be the 3 components of the cytoskeleton. Microfilaments provide shape, and then they provide cell shape and maintain cell shape. And then they also help with cell division. Cell division, okay? Actin and myosin. These are very important that you guys know that these are the 2 microfilaments, and these with muscle contraction. Overall, the function of the cytoskeleton is basically to attach the integral proteins in the plasma membrane. Intermediate filaments, these provide structural support, and they also stabilize junctions in between cells. Stabilize junctions in between cells. Okay, microtubules, these function to hold organelles, and then they also make up cilia and flagella. So those are the microtubules. So 3 types of cytoskeleton: microfilaments, intermediate filaments, and
Yes.
microtubules. All right, so our last few, and then we'll be able to move on to nucleus on Thursday. Okay. So the centrosomes and centrosols would be our next example of a non-membrane bound. So non-membrane bound organelle. Thank you guys for hanging with me. We're almost there. I know this has been a long lecture, but hopefully you're walking away feeling like you have some better detail in your notes of what to focus on. So if we're looking at a centrosome or a centriole, this is another non-membrane bound organelle. These we looked at last week in mitosis, right? They function in cell division. They pull apart the sister chromatids. Right, that was their function. The cilia and the flagella, right, like I had said just now, these are, their example is they're made of microtubules, but we want you guys to know their function. So their specific function: cilia moves things past it. So for example, mucus in the throat, it's going to move that mucus up so you can actually cough it up. Flagellum is capable of actually moving the cell. So this moves the sperm cell. Okay. And then our last non-membrane bound organelle is our microvilli. Our microvilli is not capable of motion. So if you look, cilia and flagella can move, right? Cilia moves things past it. Flagella moves the entire sperm cell. Microvilli are not capable of motion. They slow food down for absorption via diffusion in the intestinal tract. So when you eat food, this allows food to be absorbed. Microvilli are made out of microfilaments. Okay, so microtubules was an example of a cytoskeleton component, and they made the cilia flagella. So cilia flagella are non-membrane bound. Cytoskeleton was non-membrane bound, right? Centrosomes, centrioles, right? Centrosomes or centrioles, these were made by the microtubules as well, microtubules. And this again was an example of the cytoskeleton. Microfilaments, also an example of the cytoskeleton, make the microvilli. Okay, so all of these that you see on the page are really kind of part of the cytoskeleton. And just to remind you here, this page had ribosomes, cytoskeleton.